Can you guys hear me and like see my screen?
Yeah.
Yep.
Okay. So and this meeting's gonna be really short. I'm just gonna go over the rules and like an example of one or two questions and then go over what we're gonna be using for readings. So to start off, sorry about that. To start off, you are going to need goggles when needed because there is going to be a section, kind of like a lab component, but not necessarily a lab. So sometimes it can be a lab and you'll need goggles, but sometimes it just might be a diagram that you have to make with like figures to represent, for example, DNA replication or something like that. And you can use a cheat sheet. It's one page front and back. And so there's a lot of stuff you probably wouldn't want to memorize for this event. And so it's really great to make full use of your cheat sheet. And these are the topics for that we're going to go over mainly. And so the first one is polymers. The first four that we're going to go over are polymers, enzymes, bioenergetics, and cell organelles, and cell wall structure. Um, and so that's like a big chunk of this, but it's kind of some of the more basic stuff, except for so for the bioenergetics, we're not going over the bolded letters after that yet, because that is a lot more detailed. And then here are the topics for the national level. And so it's a lot more complex. And if we do get to there, then we'll definitely go over this. But otherwise, we'll go over like some of our invitationals, but like not the whole thing. And so here's the textbook that we're going to use. And I included a link to it so you don't have to go to any weird site. And this is just a direct PDF for the 4th edition. And with this event, similar to like all their events, you can have basic questions just like, what is this enzyme in DNA replication, all the way to some really complex and weird names you've probably never heard in your life. And so it's really good to just have a good fundamental understanding. And so essential cell biology is basically like a college level textbook, and it does go into a lot of depth. And so we're just going to go through it pretty slowly, so you don't have to worry about like any long readings or like any homework or something like that. Does anybody have any questions so far?
So for like homework, do you plan on assigning reading or like practice problems?
So I still haven't figured that out, but I know for example, like the homework for this meeting is just it was not problems, because I feel like we can start that later once there's like a better fundamental understanding. But just for now, the homework is just like reading a chapter at a time and um and like taking notes, but not like actual notes, but just like the most major concepts, just like summarizing them in like half a page or something.
All right, cool. Thanks.
And so here are two example questions. And so if you did take the USA Biology Olympiad, you might be like really familiar with this stuff. Um, Otherwise, like there is a lot of other studying, even if you have taken it. So does anybody have an idea of the answer for this?
Is it A?
You're on the right track.
No, because it gets transduced, right? Wait, what is a response? Damn it. Yeah, no, I don't know.
Okay, so let's just go with the question. So It's a cell signaling pathway, and C. elegans is just kind of like the most studied generic organism in biology. And the signal continues to be transducted. Our response fails to take place, so it's going through the receptor, but the response is actually not coming out of it. So it means there's a failure at like the end. And so because there's a failure at the end, and you know with G coupled protein receptors, it's an actual whole enzyme at the end and not an amino acid. You now know that it's like a tyrosine receptor kinase or something along those lines. And so it's not tyrosine. Like I know you said tyrosine because that's like the name of the receptor kinase, but that's actually like the what it's like made of. That's not how it gets transducted. It gets transducted through phosphorylation, and it usually phosphorylates threonine or serine. And so since threonine isn't an option, serine would be the best option. And you can see from the structure of serine here, there's a hydroxyl group in the R group. And so that's where a phosphate group can attach and the serine can get phosphorylated. Does anybody have any questions about that? Okay. There's also going to be like, so this is obviously something you might want to have on your cheat sheet. And it's kind of an application question. And it might even be kind of like trivia because one of these is like just kind of a random one that you might not really encounter. So does anybody have an idea for what the answer might be for this?
Maybe C. Because like it's an acid, and I know I've heard of methylene blue in like the context of indicators, but it's like a wild guess for me.
Okay, I'll let you guys like look up what each of the indicators do. Like they're all indicators, but just like look them up and tell me what you guys think.
Is it A?
Yeah. Do you want to explain why?
I just remember from last year that it had something to do with vitamin C, but I looked it up and it said it interacts with vitamin C.
Yeah. So DCPIP, when oxidized, becomes blue, and it's when it's reduced, it's colorless. And so for this, you would have like had to know in the specific level, um, or like what it's usually used for, but just like generically, since citric acid will reduce it, it makes it lose color. Okay, and so here's kind of the plan for this month. So it's just these like more basic units, and even if you take an AP biology, I would just say to make sure to like actually read through this because a lot of this stuff is much more detailed than you learn in an AP class. And so you might like think you know all this, but there's a lot of stuff that you didn't learn in AP or honors. And so it's a good idea just to read it through, especially in the later units. It gets much more complex than what you learn in school. And I will send up this PowerPoint. And does anybody have any questions?
No, I think I'm good personally. We just need to read chapter 1 by next meeting, right? And then like have a half sheet summary.
Yeah, that's just like, or just like any small summary of like the major concepts, maybe like one or two major things per chapter.
Okay. Great, sounds good.
So to answer your question, there is also a lab part to the test. It's just not always like a lab to to say like in the terms that you might know it. It might just be like like constructing a diagram of something. So it's not necessarily like a chemical lab, if that makes sense. Are there any other questions? Okay. So this was relatively short, and that's all we need to cover. So I guess that's all.
All right, thank you for the meeting, Assad. See you later.
No problem. See you.